Within just a few milliseconds, the action potential cycle and the synaptic process occurs millions of times within the brain. Since we are quite familiar with the action potential cycle, let's take a closer look at the synaptic process. But first, a short summary about action potentials to set the stage. Sodium ions are drawn into the cell because the concentration gradient is lower within the intracellular space and the membrane is more negatively charged. When sodium gets open, sodium rushes in to maintain equilibrium, which causes an immediate rise in positive ions and also causes the membrane to reach a positive charge. This sudden depolarization triggers an action potential. As an action potential travels down the axon, the electrical signal reaches the terminal buttons on which is the presynaptic membrane. Within this presynaptic membrane are synaptic vesicles containing chemical neurotransmitters. At the presynaptic membrane, a process called exocytosis occurs. As an action potential reaches the terminal buttons, the voltage-gated calcium gates open, causing a rush of calcium ions into the cell. These calcium ions assist with transporting synaptic vesicles, which again contain the chemical neurotransmitters, like items in a Ziploc bag, to their release zones and the presynaptic membrane. The synaptic vesicles fuse with the membrane and dump the neurotransmitter contained within the synaptic cleft, or synapse, the tiny space between a presynaptic and postsynaptic neuron. Within the synapse, neurotransmitters must bind to the postsynaptic receptors located on the postsynaptic membrane, usually on the dendrites of the next neuron. These receptors detect the neurotransmitters and process the information, then send another signal from the dendrites to the cell body, triggering another new action potential to start the cycle over again. If neurotransmitters are unable to bind to postsynaptic receptors, they are either taken back into the presynaptic membrane to be reused in a process called reuptake, or become deactivated within the synaptic cleft. During deactivation, enzymes break down the neurotransmitters so that they will not be able to bind to postsynaptic receptors in order for the synaptic process to be terminated. Now that we've covered the processes of a single action potential at the synaptic level, we'll move on to discussing neurotransmitters on a deeper level. To review everything I've gone over, as an action potential reaches the terminal buttons, synaptic vesicles containing neurotransmitters are sent across the presynaptic membrane and then bind to receptors on the postsynaptic membrane. If they're not able to bind, they are either reuptaken or become deactivated through enzyme breakdowns in order to terminate the process and make way for a new neurotransmitter. Now we'll discuss inhibitory and excitatory neurotransmitters. When neurotransmitters attach at the postsynaptic receptors, they can either create excitation, which increases the likelihood of the postsynaptic neuron to trigger an action potential because of an increase in positively charged ions, or it could create inhibition, which is the exact opposite, by decreasing the chance to trigger an action potential due to an increase in negatively charged ions. For example, neurotransmitters norepinephrine and glutamate are excitatory because they increase the chance and rate of action potentials triggering in the receiving postsynaptic neuron, while serotonin is inhibitory, oppositely slowing down action potential triggers. When an excitatory neurotransmitter attaches to a postsynaptic receptor, excitatory postsynaptic potentials, shortened to EPSPs, occur, usually in the form of graded potentials, which are small changes in the membrane potential but not quite enough to trigger a full action potential. This is due to a brief period of depolarization. Likewise, inhibitory postsynaptic potentials, shortened to IPSPs, experience brief hyperpolarization, also resulting in some graded potentials. This hyperpolarization is the reason why it is harder to trigger an action potential. An axon hillock, the zone where the soma and axon meet, is responsible for adding and integrating ESPSs and ISPSs. When multiple of these occur at the same time, the axon hillock integrates them in one of two ways. The first is temporal summation, where it adds up potentials that occur at the same place on dendrites over time, then sends the signals down. When temporal summation is used on graded potentials, they are instantly combined into one in order to meet the threshold of excitation and trigger an action potential. The other process is called spatial summation, where it adds potentials that occur at different places at the exact same time. When spatial summation is used on graded potentials, it can help the membrane potential from becoming too hyperpolarized to trigger an action potential. The two processes work in tandem to keep the membrane potential from being hyperpolarized over time and adding graded potentials together to trigger an action potential. So, in brief summary, the entire process is as follows. The threshold of excitation is reached, causing an action potential. The action potential travels down the axon, reaching the terminal buttons, which receives the electrical signals. Neurotransmitters are released from the terminal buttons and travel across the synapse to bind to postsynaptic receptors on the dendrites, usually the dendrites of the postsynaptic neuron. And finally, signals received from neurotransmitters are added together through summation to create additional action potentials, starting the process over again.